Okay, so today the first binaural. So what does that mean? So it's where the ears come together, two oral. So it's the first nucleus. This is the nucleus laminaris, which is which is like the medial superior olive in a mammal, MSO, medial superior olive. And so this is where comparisons are made between the left and the right ear. So, and I'll put the little bit of the anatomy up, the relevant anatomy up as we go along. So what's the overall goal? So the overall goal is to try to figure out how do we localize sounds. So how do we tell, like, in the dark a sound happens someplace? How can we tell where that sound is? Now, when you first hear about that, you say, well, that's not very cognitive. It's kind of like a low level. But that stuff like that is very important for all different kinds of higher level operations. So an example is you're in a crowded bar. There's people yammering and talking every which way, and there's music on. If you're talking to somebody in that kind of situation, the loudness of their voice is not much louder than the loudness of somebody else yelling in your ear, and yet you can focus on just one stream of phonemes coming out of the person you're talking to sometimes. So this system is very important for all different kinds of auditory attention things as well. And so there's so it's not just, it's a very low level system at the very beginning, but we'll see that at some of the information, even the frequency specific information is propagated up into the cortex. So let's just quickly put where we are in the system. So basically we've got the hair cells, the synapse on the ganglion and the spiral ganglion, and then they branch. The same exact information branches, and it goes to these little potato-like cells that have the really good time information, and then it also goes to these much bigger cells that scramble up the phase information because they've got more capacitance. And then this forms the amplitude pathway. Which we're not going to talk about today. So that's like the X-like pathway, the sustained-like pathway, and then these guys are in the time pathway. That's what we're talking about, mainly talking about today. We'll talk on Friday. I'll go back and talk about the amplitude pathway. And so here's the other side. So what are we talking about today? We're talking about getting a copy of that time information. So here's the nucleus magnocellularis on the other side, little potatoes on the other side. So getting that time information and bringing it together. So crossing, so like the left nucleus laminaris is getting input from the right magnocellularis and the left magnocellularis at the same time in order to do this comparison. So we're going to talk, so today we're going to talk about, like, what if we went with an electrode and recorded in this nucleus that is getting input from both ears. So what's the overall problem? So the overall problem is we're just trying to figure out where a sound is coming from, and there's several methods. It's like when I talked in the advanced lecture about depth perception. There's lots of different methods of depth perception. You can do the sharpness of an object. You can figure out, like, from your lens how much you've focused, and that tells you how far away it is. There's perspective, there's overlapping, there's shading, there's stereo, there's all these different mechanisms. Same way with sound localization. So one obvious mechanism is, if something is on your right side, it's going to be louder in your right ear. So if you made a comparison between the loudness of the sound in the two ears, that's one way of detecting where the sound is coming from. We're not going to talk about that today. We'll just talk about, let's try to figure out where the sound is coming from using the time pathway. And if we consider that problem, what is that problem? So if you put somebody in a dark room with a bunch of speakers behind curtains, completely naturalistic situation, right? So you just have all these speakers hidden behind a black curtain. And so these are supposed to be speakers. And then you just ask them, you play a sound out of one of the speakers, you've got speakers all over the place, and then you just ask the person to point to it. So, what happens if you do that? And let's just, consider what the problem is. So here you are, you're in the dark room, there's your nose, ears. So what's happening is the sound waves are coming out of the speaker. So here's some sound waves coming out of the speaker. And so what are those? Those are the pressure waves. So if you freeze it at a moment in time, and then plot the pressure, the pressure is, I just drew this line indicating where the maximum pressure was. And this whole thing, that whole sound wave is going about 700 miles an hour out from the speaker toward you. But you can immediately see what's the problem here? Well, if you're trying to measure, so the sound in your right ear is going to, for a given wavefront, is going to get there a little bit later than the sound in your left ear. Like here's this wavefront here is hitting your left ear. It's going to take a little while before it hits your right ear. But you can immediately see what the problem is. If we're trying to sort of like figure out which wavefront goes with which wavefront, there's a problem. We don't know, like, there's wavefronts coming by in the left ear and the right ear, who goes with who? And that's the phase ambiguity problem. Like, what is the actual offset? What is the actual phase, or the actual sort of delay, or you could also think of it as phase, offset? There's an ambiguity. Now, when you first look at this, it sort of seems like, shouldn't this be relatively easy to figure out? So if you imagine this is your left ear. Lear, no, that's no good. Okay, how about left ear? So say we just have a sound in your left ear, and say the sound does something like this. So that's the sound wave in your left ear. It's got multiple frequencies in it. And now let's say the sound in your right ear, so that was a complicated sound, like, a car going by or something like that. So here's the sound in your right ear. And so let's say the car going by was a little closer to your left ear than your right ear. So then the sound wave is going to be delayed over here. Not an exact copy, or something like that. So what's the problem we're trying to figure out? Well, we're just trying to figure out, like, what's the delay between these two things right here? That's what we're trying to figure out. And it sort of seems like an obvious problem. You got like something and there's a bump, and then there's something and there's another bump. Well, you should be able to figure that out. So what is this called? So this is called, for short, ITD. So that's interoral time delay. And or time difference, time delay, difference. So that's what we want to figure out. So what's the problem? Well, it turns out it's kind of an aperture problem. And every year when I say this, people are saying like, you're trying to explain something to me by saying it's like an aperture problem? I thought that was a problem. But it's kind of a similar thing going on. And so what's the problem? Well, we've gotten this sound, and then we fed it through the cochlea, and what did the cochlea do? It divided it up into different frequencies. And why did it do that? Well, we want to be able to tell, like, there's a high frequency thing that's different than a low frequency thing. We don't want to just sort of take the whole mess in. And so what that means is that we've created a problem for ourselves. And it's kind of a little bit like the aperture problem in the sense that, like, where does the aperture problem for motion come from? The problem comes, so if we just had to sort of like figure out whether the whole world was moving to the right, it would be pretty easy to set up a detector that could sort of detect that the whole world was going to the right. But if we make a whole bunch of teeny tiny little detectors that are looking at just a little piece of the world, then those detectors have a problem with not being able to detect the slip along the edge. So you got a little tiny drinking straw detector, and here's the thing moving, and it can't pick up this direction, and so it just picks up that perpendicular component, and then you just end up with the aperture problem. Well, this is kind of the same thing. It's analogous, except that the problem came because we divided up the world into different frequencies. And so once we've divided up the world into different frequencies, then the left ear is going to get that at a particular frequency, and the right ear is going to get something else. And then the question, another copy of it. And then the question is, does this wavefront, sorry, I didn't make the frequency the same. Now let's try to make that frequency the same. There we go. So does this guy, correlate? Is that the delay, or is it that delay? Say this was the left ear and the right ear. So was it earlier in the left and quite a bit later, or was it maybe the other way around? Maybe it was maybe the right ear came first. And so this is the phase ambiguity problem. And what causes it is the cochlea, basically. Because we went through the trouble of filtering things into different frequencies, sort of similar to the visual system where we went through the trouble of making a whole bunch of little motion detectors that are just measuring motion very locally. Because of that, then we end up with this, we've created this problem for ourselves. And so today, how do we solve this? And so it's a little complicated, so we'll do the first half of the solution today, and then on Friday we'll sort of figure out how it works. Now, when you sort of see this stated, you think, well, it's kind of, it sort of seems like an abstract problem, but is it an actual real problem? And yes, it is a real problem. This was figured out many years ago by like 80 years ago by psychophysicists, auditory psychophysicists doing experiments with speakers. And so if we make if you go in a room, I actually have been in one of these experiments. You go in a room and you got the little black curtain, lots of speakers all over the place, etc. And you play a sound out of there. So if you play a sound that has a lot of frequencies in it, like a psh kind of sound or a sh, that's a sound that contains, it's generally high frequency, but it's got a lot of different frequencies. If you play that sound out of a speaker, you can localize it really easily. You can immediately just point to where the speaker is. But so if you have multiple so you're sitting here, there, ears. So multiple frequencies, no prob. You can very easily tell where the sound is coming from. But if you play a pure tone, that just means one frequency. It turns out it's quite hard to localize it. Now, luckily, the world doesn't have very many pure tones in it, or at least it didn't before computers came along. So a tuning fork makes pretty much a pure tone. So if you get a tuning fork and ding the tuning fork, that will pretty much make a pretty pure sine wave. This is a physical thing that will make a sine wave. And if you do that, you can see, if you get a tuning fork and sort of ding it, close your eyes, or have somebody else ding it, and then put it somewhere, and then try to figure out where it's coming from. It's got this weird sort of ambiguous sound to it. And so if you do that, if you play a pure tone out of one of these speakers in the dark or with the curtain, you might say, yeah, I think it's coming, and then you play a pure tone and then ask there are different ways of doing the experiment. You could say, was it coming from this speaker? And then the person will say yes. And then you ask, was it coming from this speaker? They'll say no. But it turns out if you play it out of this speaker and then ask, was it coming from this speaker? Then the person will say, oh yeah, I think it was coming from that speaker. And if you go and play it out of this speaker over here, it's completely different than this one. So play it out of this speaker and then ask, was it coming out of this speaker? The person will say, yeah, it's coming out of that speaker. And so there really is a problem with a pure tone. And for many years, they used to make these backup alarms that were pure tones. And just like a beep. And so for many years in class, I would say, you backup alarm guys, you got to learn more auditory psychophysics. You shouldn't use a pure tone because that's very hard to localize. And so over the years, they've actually improved. And so now backup alarms are more like psh kind of thing. They got a little beep in them, but there's more frequency, so you can localize them. So you end up with, when you do a pure tone, you can localize it, but it's periodic places, because you're basically doing this. Periodic places, so it's not coming from here, but you can't tell whether it's coming from here, or here. So if you look at animal evolution, animals have figured this out. So if you look at alarm calls that animals make, so if you have a chicken there and you make like a little shadow that looks like a hawk over it, the chicken will give out an alarm call to warn the other chickens to run into the brush because there's somebody dangerous around. I see in my backyard whenever the hawk arrives, all the birds take off. But they also send out alarm calls. Now, if you think about that, that's kind of a dangerous thing. If you're sending out an alarm call, you're telling the hawk where you are. And so what have birds done? When they send out an alarm call, it's a pure tone, hard to localize. So they specifically make a nice, clean, pure tone when they send out an alarm call. And this happens with other animals as well to make it hard to localize. So it actually is a problem. And so let's now start to look at what the responses from the nucleus laminaris look like. So if you go into a cell in the nucleus laminaris and just record, so this is nucleus, laminaris. So he's going there, and which is kind of like the medial superior olive, MSO. And so this guy it's binaural. That means that it gets input from the left and the right, but it's still frequency tuned. It has a map of different frequencies in it, and so the neurons have to respond to a particular. If you don't play them the right frequency, they'll just ignore it. So if we plot the interaural time difference along this axis, so this is, so say here's 0 here, so this is I, T, D, so it's this guy here, interaural time difference. Now, what are the units of this? So that's an important point here. So the units of the interaural time difference, they're pretty small amount of time. So consider, like, if we go back to this picture here, so here's a sound wave. It hit the right ear, but it's going 700 miles an hour, and so it's a very small amount of time later, this guy will hit the right ear. If it hit the left ear first. So the units here are microseconds. So a microsecond is a millionth of a second. So let's plot a bunch of different. So here's 50 microseconds. Say, how do we get up? Yeah, 100, 150, 200. Okay, so those. So what's 100 microseconds? So 100 microseconds is a tenth of a millisecond. So a millisecond, that's sort of more easy to remember. So a millisecond is a thousandth of a second, and so a tenth of a thousandth, 1/10,000th of a second. That's a very small amount of time. You notice it's actually narrower than a spike. A spike is about 1 millisecond, and so this is like 1/10 the width of a spike. It's a very small amount of time. So if we go and just play a bunch of sounds to the left and the right ear, and so how is this done? People made some owl headphones. So you can't get them on Amazon. You got to make the owl headphones, and they played these sounds with different time delays, and so this is right ear leads, and the other side is the left ear. So 0 is like if an object was right in front of you, because it would make the same time. So right leads, left leads. So if we just record the response of the cell, what happens is you'll find, say, a cell. So here's the 0 mark. Say you do a bunch of recordings and you find here's the response of the cell. So here's the response. Like spikes per second. Spikes per second. So you do it multiple times. You get some for a particular interaural time difference. You get some responses, and then if you turn up the time delay, make the right ear even earlier relative to the left, you'll get another response. And if you keep going, at this point I always say, so in the what do we have? We've got a curve like that, and then we have some sine waves. So everything is sine wave. So this is going to be a sine wave, but it's going to be a sine wave of responses as a function of interaural time difference. And so it looks like this guy is sensitive to interaural time difference. So you can see that if if you keep making the interaural time difference bigger, the response goes down. But then if you continue bigger, it goes back up again. So you can see, and each one of these, you would sample at a bunch of different interaural time differences to figure this out. So basically you get like this sine wave of different responses to different interaural time delays. So you end up with this, yes, one last time. It's very hard to draw a sine wave of a particular frequency. There we go, I think I got it that time. Okay, so what does this mean? So what this means is, if you get a big response out of the cell, so here's a big response out of the cell, and here's another big response out of the cell, and here's another big response out of the cell. So you can't tell whether, just because you got a big response, is it actually this time delay? Like, was it actually 30 microseconds? No, maybe it was like, minus 140 microseconds, or maybe it was, 200 microseconds. So this shows you that the cells in the nervous system, in the nucleus laminaris, they have this problem. They really can't tell. They've got some information. They say, I know it's not minus 50. So they can tell for sure it's not minus 50, but it could be like, minus 130. So it's an important point. Some information has been extracted from the signal. It's just that it's ambiguous. So basically, this is, ambiguous. If you get a big response, big response is ambiguous. So you can't tell whether it's that, or it's that, or it's that. So this problem does exist in the nervous system, very similar to it's the same thing as here. When you hear a pure tone, you can't tell which. It's not here, but it could be at these other locations. So what happens if we go to another cell at the same frequency? What you'll find is another cell will have an offset response. It'll also be ambiguous. But this guy is going to like, say, 60 microseconds, and this is supposed to go a little bit further on, and it's going to like, say, 250 microseconds out here. And it might, instead of liking minus 130, it might like minus 70. So it's going to have a different set of ambiguities. So this cell is going to know for sure it's not, it's not 0, but it could be like, minus 70, or maybe it was, plus 70. Can't tell. So how does so what's causing this? So what's causing this in the nucleus laminaris? So now we'll go back to the anatomy. So we've got the nucleus magnicellularis. That's the monaural nucleus. That's the phase-locked one. That's indicating the peak of the sound pressure waves. And then the nucleus laminaris is the first nucleus where they come together. So what causes this ambiguous response? So here's let me clear this out of the way too. So if we just draw the nucleus angularis and nucleus magnicellularis, so here's our nucleus magnicellularis. This picking up the little potato cells. And then here's a nice big nucleus laminaris. And then here's the midline. So there's another nucleus laminaris on this side, which we're not paying attention to. So here's another one over here. And then there's another nucleus magnocellularis on this side that we are paying attention to. Okay, so this is nucleus magnocellularis. Here's the other one, nucleus magnocellularis, right, left. And then this is the nucleus laminaris, and left. There's a right one also, but I'm not drawing it. Okay, so what are the connections? So the connections were here's a cell, and it comes in to the nucleus laminaris and runs across it, like that. Now remember, each one of these things there's a frequency map here. So frequency is going in this direction. So different cells in there respond to different frequencies because they're coming from different parts of the cochlea. And there's a frequency map in this guy too. So when the cells here project to here, they just go to a particular little frequency part of the nucleus. So if you look at a different cell here, that different cell is going to come in and go to a different part of the nucleus. There's a frequency map. So how about the other nucleus magnocellularis? This is the one that's breaking the rule of Serino because it's crossing over to the other side of the brain without crossing the pons midbrain junction. So this guy will, same part of the nucleus, will come out and then run over to the other side and run across the nucleus in the opposite way. So, and the idea that this is how it works, that's the thing about humans, tricky primates. They actually figured out how this worked basically long before they actually had the evidence for it from nucleus laminaris, or the anatomical evidence I talked about. There was a guy named Jeffress who kind of figured this out. Jeffress, can't remember, might be one F. Yeah, I think just one F. Jeffress. And so how does this work? Well, this little potato cell comes and it makes a whole bunch of synapses. So each one of these are synapses, like that. And then here's the actual cells in the nucleus. So here's the nucleus laminaris cells. So there's a whole bunch of them like that. They're kind of a little potato-like as well. And then the ones coming from the opposite side, they make synapses on the potato cells also. And so basically what you have is a coincidence detector. Got those synapses a little far away from the cells. There, we'll make them touch the cells. So how does this work? So basically what it does is it just detects when there's a coincidence between two spikes coming in the opposite direction. So if we put a spike here, it's a little wider than this, but I'll just draw it here like this. It's coming like this, coming down the pike. And then let's have a spike in the other, on the other side. So here's a spike right here on the other side. This guy's, coming down the pike also. And so what happens is they'll come into the nucleus, and then they'll, run across. Let's put this guy slightly further away. Put him slightly further away. This guy was about that far. Let's put him about over here. This guy's a little delayed. So it's coming that way. So what will happen, they'll sort of run down like this, and this guy's a little delayed relative to this one, so this one will get across the nucleus a little faster, and so maybe they'll end up coinciding at that nucleus, at that cell, that nucleus laminaris cell. And so that will mean that, so what's happening here, well, the spike comes in and it causes all these guys to release neurotransmitter with a little delay going across the nucleus, and the same way the other side. And so since this guy will go off at the same time as that one, that neuron can detect, I got input from both ears, and then fire. But you can see what the problem is. So the problem is, how do you figure out which spike to match? So these are two separate nucleus magnocellularisuses, and so they don't have to make all the same spikes. We said that, if you've got a sound like this, it might spike on that peak, but it might skip one, and then it will go and then spike on another peak. So what if this nucleus laminaris was just kind of tired? It didn't feel like spiking on that one. That can happen because they're just two separate streams of information. And so now we got a problem. So what does the nucleus laminaris do? It just matches any old spikes. It doesn't know which one matches with which. And so say there was another one here that was a little bit further advanced. So there's another spike coming down. Well, what's going to happen? Well, that spike is also going to match. It will match, at a later point in time, like maybe it'll match over here. So these guys will come into the nucleus and then they'll match here. So basically these two spikes will come in. If you have two spikes on one side, they'll come in, and what will happen is this first spike will match here, but then a little bit later in time, the same spike will match with this spike. So basically what the nucleus laminaris does is it just matches spikes. It just matches spikes, and it doesn't know, are we supposed to match those spikes? It matches any old spike coming down the pike, because it can't tell that this spike was the real one, the real delay. The real delay could be this, and so that's why you get this. That's why you get this kind of curve. So basically, what happens is, so this was matching, say the first spike, and then if that one is earlier, this one was matching the second spike. So you only got one in the left. Now it gets more complicated in real life. You could have like two spikes that weren't evenly spaced. Like what if, this came down, like this came down one side, going that way, and then something like this came down, the other side. You could have lots of matches. As these pass, you'd get a match there, and then you would get a match on these guys, and then you would get a match on, these two. You'd end up with 4 matches if you just have 2 spikes coming in each side. So it's kind of confused. But now the question is, it's done something. We've made some progress because we've got another dimension here. And so what's this other dimension? This other dimension, if you read, like if you go and read one of the articles in the reading, it'll just sort of be quite confusing. So what is this one called? So this one, when I first heard this, I watched talks from the Kanishi lab. Mark Kanishi is one guy who sort of figured out a lot of this stuff in the owl. So this axis is called the characteristic delay. So that's the characteristic delay going that way. So it just doesn't do it for me. Like characteristic delay, what the heck does that mean? It doesn't make any sense. But it's got some information in it, because if we look at a different cell in somewhere along that same frequency spot, we're going to get an offset curve. So it's mapping something. It's another ambiguous curve. This curve is ambiguous also, but it's done some work, so that you can see that different parts of the nucleus are representing something about the delay. So that's the problem. So how on earth could you actually figure this out? Now there's one more little tricky thing, given that this is biology, we'll use whatever we can. And so if you actually look at the axons here, so if you just look at this axon here, what does it look like sort of microscopically? So normally what happens, you talked about myelin, and myelin has the property of reducing the capacitance, and when it reduces the capacitance, it makes the action potential go faster, because you don't waste any time charging up the capacitance. So what happens here? Well, it turns out there's a whole pile of nodes of Ranvier. So what happens is the nodes of Ranvier there's like tons of them. So that's supposed to be the myelin. So this is a node of Ranvier where the myelin goes away, and then there's channels exposed, and so some channels can actually conduct. Now, normally, if you want to make the axon fast, you have like a long distance between the nodes of Ranvier, so that you can get the fast conduction down to the next node. But what the owls do just here in the nucleus, they increase the number of nodes of Ranvier to slow the conduction down. And so that's supposed to be the myelin, node of Ranvier. This is supposed to be the myelin. So they actually slow it down. So how does it work in real life? Well, you've got these axons, and they whip down these part of the axon, which has widely spaced nodes of Ranvier. And then when they get into the nucleus, they slow down, and so you can do the matching better. So you can kind of spread out the matching a little bit. And so they purpose, and how on earth do your genes specify that this should be the case? That's not understood, but somehow the nervous system knows to make more nodes of Ranvier here in between the synapses. The synapses, that's just the axon, but the synapses, will come off here. So a little axon branch will come off, and there'll be a little synaptic bouton like that, just coming off of the node. And then this will be like, potato. That's a potato cell right there. So, yeah, just so that's how the actual connections come off. But the thing is, so they whip down like this and then go slowly through there, so that they can match all these coincidences. So how could you solve the problem? So what we looked at here was just, how to solve a problem. So what we looked at here was basically just one frequency. We looked at a bunch of different cells, nucleus laminaris cells. So this is, I should label this, different NL cell, same frequency. So if we just had one frequency, we can't solve the problem. So we have to look across frequencies. And pretty much it's like the aperture problem all over again. What is looking across, how do we solve the aperture problem for pattern translation? What we did was we took all these ambiguous samples from little pieces of the object, and then we looked across space in order to figure out what is the pattern motion that's most likely. So each detector is ambiguous. So if you have like this object like this, so this detector thinks it's going that way, this one thinks it's going that way, this one thinks it's going that way. So there's all different possibilities. Each one of these, you don't know, it could be going that way or that way. So each one's ambiguous. So each one of those guys has a little, family of different pattern motions. And how do you solve the problem? You look across all those detectors and find out which pattern motion is the most consistent with the data. And that's pretty much how you solve the problem here. So you have to, combine across frequencies, which means sort of combine, across the frequency map, which you're not doing here. So nucleus laminaris just does one frequency at each point in the nucleus. So you have to combine across frequencies. That's, like, combine across space. Space for, aperture problem for pattern motion, pattern translation. So you have to combine across space. And so roughly how does it work? We'll talk about it in more detail next time, but roughly how it works is if you have one frequency, the cell is going to give you a response as a function of interaural time difference that says it could be that one, could be that one, could be that one, don't know. But if you've got another frequency there, so what is the, if we go back to this diagram, what is this, what is that difference, what does this difference mean? So let's just see what that difference is. So what that difference is about, say it's about 200. It's a little bit less, but just 200 for easy math. Say it was about 200 microseconds. So if that's 200 microseconds, what is that? What is that time? That time is just the time between two sound pressure. So now this is time, not ITD. So here I've plotted ITD, which is the time. So that's left. It's not time. So it's left versus right difference. That's what I'm plotting there. That's why I said everything looks like a sine wave. Okay, this is an actual sine wave of sound coming through the air. And so what does this amount of ITD represent? It just represents the time between two of these peaks. And if this is, say, 200 microseconds, what is the frequency of a sound that's 200 microseconds between peaks? Well, if it was 1,000 microseconds, that's 1 millisecond between peaks, that would be 1,000 peaks per second. So 200 microseconds between peaks means it's about 5,000 cycles per second. So that's what the meaning of those peaks are. So if we go to a different frequency cell, a different part of the nucleus, what's going to happen is we'll end up with differently spaced peaks. So this cell will say, I'm not sure, it could be that interaural time difference, or maybe it's that interaural time difference, or maybe it's that interaural time difference. And so now you can sort of see how the problem is solved. All we got to do is just find the interoral time difference that has the most votes for it. So this, this interoral time difference has 1, that guy just has 1, but this guy has 2. And so that's probably the real one. So that's not done here. It's not done here. All this, so this is, it's important to sort of, get the order of things. This is just the high performance input thing that's just matching spikes. This guy's just say, I don't know whose spike is who, I just match everybody. And let somebody else down the line figure it out. And so then down the line, in order to figure out what the real delay is, you've somehow got to do this kind of aperture problem. And so what is that, that aperture problem? Well, it's just, find one delay, ITD, interoral time difference between the two ears, that all, I'll just sort of write it out very verbosely, that all different frequency ambiguous, so each one is ambiguous at each frequency, different frequency responses all agree on. So it's kind of like, it's like the aperture problem for pattern translation. It's not a, it's not a perfect, it's not a perfect answer. There's going to be a somewhat ambiguous answer. There'll be votes for all different pattern motions, but one of them is going to win, have the most votes for it. And so it's the same thing going on here. Okay, usually I kind of whip over this in about 20 minutes, and so I went over it longer, so hopefully it was a little clearer than in my bad old days. So any questions? Any questions about the nucleus laminaris? So the same thing's going on over here, but it's for the other half of space. That's the other nucleus laminaris. So any questions? Yeah.
It's not really a question, or just like a question for thought. When you talked about the phase ambiguity problem, you said that like when multiple frequencies are presented, like we don't really have a problem with it, but that a pure tone like is what we kind of have issues with. Do you think that's like an evolutionary thing because we don't naturally, like you said, like hear a lot of pure tones like in like our everyday life until technology kind of became a thing? Do you think that's like has to do with evolution?
Sort of. I mean, I think the question was, does this phase ambiguity with pure tones, is because that there weren't pure tones. Well, I told you one example of like with the birds, they made a pure tone. They evolved a pure tone so that they could, not make themselves a target. But no, I think the pure tone business is really more to do with the fact that the cochlea essentially makes almost pure tones. So it actually just, so in order to analyze sounds, we need to somehow, get signals from different frequencies. But in the process, you generate an aperture problem because you're essentially a given, hair cell is only sensitive to a particular frequency. And so that's really where the problem comes from. As soon as you've made some kind of device that detects different frequencies, you create this problem if you're trying to detect the delays because then you can't tell which peak goes with which. Whereas if you looked at all frequencies, you'd be able to look at the overall shape of the sound wave and say, oh, I can easily see that guy is offset by, by so many microseconds. So I think it's really, it's just the only way you can do it. If you want to measure different frequencies accurately and have different signals for different frequencies, then you're going to create this problem. So is everybody pretty clear on sort of the matching thing. So basically what happens, the spikes come in and they cause every single one of these synapses to fire. And then if only one synapse fires on the cell, if there's some kind of threshold, the cell won't fire. So it will actually require two synapses going in the opposite direction to fire in order to make that cell. And then this cell will go off and onto the inferior colliculus, central nucleus, lateral part. And that's what we'll talk about next time. Yeah. Oh, the character. Yeah. So that's an important point. So what you've done when you set up these lines, you've got something being mapped this way. And so what is it? It's basically kind of the offset of the ambiguities. So you either have one cell will say it's 25 or minus 150, and the other cell will say it's 75 or minus 70. So it's mapping something. It's kind of mapping a real, it's mapping a real delay in how long it took the spike to go across the nucleus. But and you need these multiple parts of the nucleus to make this work. I'll try to make a diagram next time of what the activity across the whole nucleus looks like. So you need characteristic delay in order to make this, to make this work. So if you just had one single coincidence detector that was, say you just had, one cell, and none of these other guys with different characteristic delays, you just end up with one of these curves. And it turns out that's not enough information to solve this problem. So, I mean, it's a real delay because it just reflects, the delay it takes to get across the nucleus. But hopefully next time when we look at if we imagine here's the entire nucleus and here's all the parts of the nucleus that are activated, it'll become a little clearer. But it's the way the nervous system always makes a map of something. So it's made a map of this offset of these ambiguities. So what is it? It's it's a map of the offset of these ambiguous responses. The different, as you move across the nucleus this way, it just shifts this ambiguous curve across interaural time delay. Yeah, so it's a confusing thing. I don't know if there's a better word for it. Yeah. Yeah. Yeah.
Okay, so there are regions of multiple nuclei, even though it's just called nucleus.
Yeah, there's a bunch of cells within here. Yeah, so if you go in nucleus magnocellularis, there's a whole bunch of cells, and you go into different parts of the nuclei, they respond to different frequencies. Same thing over here. So this is frequency going this way. So as you go to different parts of the nucleus here, there'll be another cell down here, and this one will respond to, say, higher frequency than this one. And so if that responds to higher frequency, that's going to make the ambiguities change, because this distance here is basically just the time between two successive sound waves. And so if you've got a higher frequency, there's going to be less time between the sound waves. And so you'll end up with a whole pile of different ambiguities, because they'll be closer together for a higher frequency sound. So it's a two-dimensional. It's actually, there's the third dimension, so the nucleus extends out like that. But remember, we talked about those planes coming in. So there's kind of like a plane of these things. Actually don't make lines. They actually make a little plane of synapses across here. They come in like this, and they synapse on on the whole plane. So it's actually three-dimensional. But if we just forget about that dimension, there's two main dimensions. So one of them is frequency. So cells, as you move in this direction, they respond to different frequencies. As you move in this direction, they respond to different characteristic delays. Yeah, so this is kind of a little bit of a technical thing you don't usually go over, but I think it's interesting just to see how the nervous system it's just like figuring all these things out, just like trying all little tricky things to sort of figure out, creating problems for itself and then solving these problems, and all in the service of not getting eaten and having a good life. Okay, so next time we'll talk about how do you actually sort of construct a map of space out of this, and then also how do you actually use this information to direct auditory attention to different stuff in the room. Okay, I'm way over again. So, see you guys.